In this next section, we'll see how the FDA's current role, scope, and function has evolved. So, what does the FDA regulate today? FDA is responsible for protecting the public's health by assuring the safety, effectiveness, and security of human and veterinary drugs, vaccines, and other biological products, medical devices, our nation's food supply, cosmetics, dietary supplements, and products that give off radiation, as well as regulating tobacco products. In fact, FDA oversees items accounting for 25% of every dollar spent by consumers. You might be asking, what does the FDA not regulate? Quite a lot, actually. As the slide shows, the FDA doesn't regulate advertising of over-the-counter drugs. Alcohol or drugs of abuse, that is, non-medical uses of controlled substances. Nor do they regulate government health insurance for the elderly, disabled, and those with low incomes. The FDA doesn't regulate meat and poultry, pesticides, or restaurants and grocery stores, and most other household goods, nor does it regulate public water supplies, although it does regulate bottled water. There are many ways in which the FDA routinely seeks input from people outside the agency. Aside from the expertise of its own staff of physicians and scientists, the FDA can also talk with external consultants, either on an ad hoc basis or as members of its advisory committees and panels that meet several times each year to discuss difficult or controversial cases and emerging issues. Increasingly, advisory committees have patient representatives at each meeting who are trained advocates for people with specific diseases or conditions. In the Center for Drug Evaluation and Research, CDER, patient representatives serving on advisory committees have a vote, while in the Center for Devices and Radiological Health, or CDRH, patient representatives serve on advisory committees but do not vote. Advocates can serve as patient consultants, participating with FDA staff in closed meetings with pharmaceutical companies concerning the design of clinical trials. The opinion of the public is directly solicited at the open public hearings that are a part of every advisory committee meeting. The FDA also seeks comments on draft guidances and policy documents, keeping a docket, that is a place to record comments, on their open, publicly accessible website. Public comment is taken seriously. Maintaining the greatest possible transparency about its policies and process is important to the agency. Let's define some terms used by the FDA. When a new drug is approved, it is for a specific indication. Indication is the term used by the FDA to refer to the use of a drug for treating a specific disease. The research must demonstrate what the FDA refers to as a clinical benefit. In cancer, for example, the FDA defines clinical benefit as prolongation of survival or an improvement in symptoms for that indication. In approving some drugs, the FDA has also assumed clinical benefit from effects on what are called established surrogate endpoints. A surrogate endpoint is an intermediate measure that is believed to reliably predict a clinical benefit. A surrogate endpoint can be used for accelerated approval pending confirmatory trials that demonstrate actual clinical benefit using a real endpoint or outcome. However, there are questions about whether surrogate endpoints, in fact, predict outcomes important to patients. Other than death, endpoints are usually highly specific to a disease, requiring some knowledge of that disease to understand the drug approval process. Since the example we're using in the next section of this module refers to cancer, it's important to understand some of these endpoints. CR, or complete response, means no measurable tumor remaining as a result of treatment. PR, or partial response, means tumor shrinkage of at least 50%. In general, response or tumor shrinkage is not considered a surrogate for clinical benefit because it does not predict overall survival or improvement in symptoms. You may recall the example of bone marrow transplant for breast cancer, where ultra-high doses of chemotherapy produce greater tumor shrinkage, but didn't lead to better survival and certainly not to fewer symptoms. Stable disease, or SD, is used to measure metastatic cancers which usually progress or worsen over time. Progression-free survival, or time to progression, refers to the length of time from initiating treatment to cancer getting worse. Both of these can be subject to biases in measurement since they depend on being able to detect the disease in the same or a new location and the timing of this detection. The gold standard in cancer clinical trials is overall survival, or OS, a measure of how long the patient lives that takes into account both benefit and risk or harm from the treatment. Trials that measure overall survival take longer and require more participants to show an effect than trials that use surrogate outcomes. Now let's review the stages of drug development, paying attention to the role that the FDA plays in the process. The earliest stage of the drug development process involves the discovery of new molecules and chemical agents and their metabolites, often involving screening of thousands of potential candidates. Likely drug candidates are then examined in the lab for their impact on cells, tissues, or other biological markers of a given disease. Then they are tested on animals. Animals like mice are used because they share almost all their genetic structure with humans, so they're often, but not always, a pretty good predictor of a human effect. However, the FDA often requires animal studies from one rodent and one non-rodent species before human trials. Longer-term animal studies of drug safety continue while the drug is being tested in trials. Once the initial animal and laboratory studies are completed, an application to the FDA can be made to study the drug in human beings. This is called an IND, or Investigational New Drug application. To allow the company to conduct studies in people, the company must submit information on animal pharmacology and toxicology, manufacturing information, and complete plans for clinical trials, known as protocols, and information about the trial investigators. Institutional review boards, or IRBs, groups of stakeholders at the involved hospitals or academic institutions where human volunteers will be enrolled in studies, must approve the trial design prior to testing in human beings. While they review and advise, the FDA does not specifically approve clinical trial protocols. During the conduct of clinical trials, the FDA offers valuable technical assistance to medical researchers and manufacturers, helping them design better studies and resolve problems with the production of investigational agents. The boundaries between the different phases in clinical trials and drug development are really quite fluid. We're naming them here to present a sequence of testing, not to define hard and fast rules. The way trials are conducted has changed over time and varies by disease area. In many fields now, for example, most trials have a comparison group, regardless of the phase of testing. The purpose of a Phase 1 study is to find a safe and active dose, as well as to determine safety outcomes. Phase 1 studies are typically small and test different dosage levels with a few patients at a time. Patients with related diseases may be tested if the company is trying to decide on which indication to pursue. Phase 1 studies can be controlled and are not designed to test efficacy, although once in a while the effects on a majority of patients are immediate and dramatic. Phase 1 studies pave the way for Phase 2 studies in a larger group of patients, where safety is further assessed and favorable signs of drug efficacy are used to determine whether a company will go ahead with a much larger and very expensive Phase 3 trial. The FDA reviews and advises on each trial protocol, and the company must submit all trial data to them. It's during Phase 2 that issues in dose finding, impact of the investigational agent in different stages of disease, selection of endpoints, and quantification of treatment effects are focused on. In Phase 3 trials, patients are randomized to receive either the experimental treatment or the standard of care for that disease or condition, which may be a placebo if there is no recognized standard of care. This design minimizes most forms of bias. The drug company works out with the FDA ahead of time that they are planning one or more Phase 3 trials for a new drug, and as part of this conversation between the drug company and the FDA, the primary and secondary outcomes or endpoints of the trials must be pre-specified, agreed upon in advance, before the trial is initiated. Generally, the FDA requires 2 adequate and well-controlled trials to establish efficacy. This is because with a single trial, as you may recall, there is still up to a 5% likelihood that statistically significant results may be due to chance. With more than one such trial, it's far more likely that a positive result is genuine. These Phase 3 studies will be done in hundreds to thousands of patients, and the larger the study, the smaller the benefit that can be detected. Less common side effects and other safety issues will appear in these larger studies if the duration of patient follow-up is sufficiently long. When these trials and all the other data are submitted to the FDA, the company applies for an NDA or new drug application, or a BLA or biologic licensing application to permit them to market the drug, which must be shown to be safe and effective. The FDA considers both benefits and risks of the new agents, with a focus on whether the benefits are meaningful to patients and how willing they may be to accept the risks. The package insert or label must be appropriate and complete, and the manufacturing methods and controls must be shown to preserve strength, identity, quality, and purity. After the company has applied for and won marketing approval for their drug, post-marketing studies will be done by the company and other researchers to further refine the best use of the new drug in the real-world setting of physician practice. The FDA and other regulatory authorities commonly ask for post-marketing safety surveillance of new drugs. But under the 2007 FDA legislation, the FDA now has expanded authorities to require post-marketing safety studies, safety-related labeling changes, and risk evaluation and mitigation strategies, or REMS, under certain circumstances. This complex schematic drawing from the FDA offers a glimpse of the whole process of drug development, from preclinical synthesis, purification, and animal testing on the left, through the various phases of clinical trials to the FDA review on the right. The pink triangles at the base of this figure show the submission of the IND, or investigational new drug application, and of the NDA, or new drug application. If the NDA is approved, the sponsor, how the FDA refers to the drug company, can then market the drug. In the past, FDA was often faulted for taking an unreasonable length of time to review new drugs prior to approving them, but as this figure shows, preclinical research and clinical trials represent by far the largest part of the 10 to 12 years that it takes to develop a new drug. Most people assume that when their doctor prescribes a medication for them, regardless of why they are taking it, the FDA has approved the drug as safe and effective for that use. But this isn't necessarily the case. The FDA approves a drug and reviews the research only for specific indications of a drug, that is, for a particular disease or condition. That indication and the clinical trials supporting that particular use is described in the product labeling or package insert. A pharmaceutical company is allowed by law to market a drug only for its approved indication. To market to other kinds of patients or for other diseases or other stages of a disease, the company must conduct further research and submit it to the FDA for approval of a new indication. However, FDA has no legal authority over the practice of medicine, and once a drug receives an FDA-approved indication, doctors may prescribe it for other uses. This is referred to as off-label use, and it is very common. While one hopes that off-label use is supported by evidence of safety and efficacy for that condition, this isn't always the case. According to a 2006 Stanford study of office-based physician prescribing practices, the highest rates of off-label use were for anticonvulsants, 74%, antipsychotics, 60%, and antibiotics, 41%. Overall, 21% of prescriptions for the most common medications were off-label, and 3/4 of these had little, if any, proof of efficacy to support the off-label use. Some of the real-world examples we've used in our web course on evidence-based healthcare illustrate how widespread and problematic off-label prescribing can be. Perhaps the most striking example would be long-term postmenopausal hormone use in older women that was given off-label because it was thought to reduce the risk of heart disease. While drug companies are prohibited from marketing off-label uses to doctors, widespread distribution of medical journal articles by pharmaceutical reps favorable to off-label uses and other related tactics have resulted in heavy fines in a number of instances. The problem is that what doctors receive is biased toward the company's product. Treatment guidelines intended to represent expert medical opinion on standard of care often discuss off-label uses of drugs. As we've said before, FDA approval is based on only limited information available at a particular point in time. Because the public wants and expects new medical treatments to be available as soon as possible, a compromise must be struck about how much evidence is enough. As a result, safety and efficacy data are often very limited at the time of approval. A drug will have been studied only in the short term, in 1 or 2 randomized trials, in a very select population that doesn't represent the general population of patients. For example, those with other health problems or taking multiple drugs. More will be learned as the drug goes into wider use and is studied further. For example, in women, different age groups, and racial or ethnic minorities. Some toxicities will inevitably emerge after marketing, and some drugs won't work as well as initially thought. The only remedy for these limitations would be more clinical trials prior to approval that last longer and involve more patients. But the public is eager for new treatments, and the drug companies want to prolong the market exclusivity of patent protection for as long as possible.